Sarah Mikesell with the Poultry site, and today we're here with Jessie Stellberg. She is a Microbial Genomic Scientist with Arm & Hammer Animal and Food Production. Thanks for being with us today, Jessie.
Thank you very much for having me.
Very good. We're here at the Arm & Hammer Research and Innovation Lab in Waukesha, Wisconsin, and we're inside one of your labs here, and we want to talk about genome sequencing. So let's start at the beginning. Kind of what is genome sequencing?
So genome sequencing is going to be a type of technology that we use to determine the sequence of nucleotide bases within an animal or a cell's genome. So this is your DNA, this is figuring out what genes you contain, this is going to determine everything about that organism, what proteins you make, how you're going to respond to stress or other environmental factors, many, many things. So this is a really useful tool when trying to characterize these bacteria in these facilities.
Very good. And how do you sequence genomes here at the lab?
So we actually have two different types of sequencing technologies here in this facility. So the first one is going to be probably the most common technology at this point in time, which is next generation sequencing. So this is an incredibly accurate form of sequencing. It's been around for about 20 years now. You're going to find this in many labs across the country, and it's going to give us a really high degree of accuracy when looking at these genes and even single nucleotide differences between strains. The second type of technology that we use is going to be long-read or nanopore sequencing, which is actually right here. So this is the sequencer in itself, and this allows us to see more of the overall structural variation in the genomes. So we can determine if a certain gene is on the main chromosome of that bacterium, or if it's going to be on a plasmid, which is a more mobile genetic element that can pass back and forth between strains.
Very good. And then tell me how you take that information and put it together.
So we'll actually, when we assemble the genome from this data, we'll use both the accurate data from the next generation sequencer and the structures, kind of a scaffold from this smaller sequencer here. So then we can determine both the gene sequences and where they're placed relative to each other.
Very good. So it's really a combination of two pieces of data that you're pulling together, right?
It is, yes, and it helps us make a very complete picture of that genome.
Very good. And why do you use genome sequencing here at the lab?
So we use genome sequencing here for two main reasons, one of which is going to be taking a closer look at our proprietary probiotic strains. So these are the strains that are going to be in all of our products here. We use them for poultry, swine, even a few human products, and we want to make sure, first of all, that they are what's known as a clean genome, that there's no hidden antibiotic resistance genes, there's no hidden toxins, anything like that could potentially harm the animal. And then we also want to take a look at, what's making this bacterium work so well? Is it producing products that are going to help fight off other E. coli or Clostridia or bacteria that can harm the animals? Is it producing products that will help facilitate the immune system of the host animal and improve its ability to fight off its own infections or, things like that? So it's really useful to know that information about our own strains. And we'll also use it to take a look at more virulent or pathogenic disease-causing bacteria. So we can do this either in a larger scale study where we'll look at hundreds of genomes that are either associated with disease or not. Maybe these are strains that have come from an actual sick bird or cow, and then strains that have been found in healthier animals, and compare them and see if there's any genes that, kind of make up that difference. And then we can use those to make new tests here in the lab, because genome sequencing, while an incredibly useful tool, is a little bit more expensive and time consuming than some of our other tests. So if we can use the data from this to make a more high throughput test that can more cheaply and quickly look at a lot of strains from each of these facilities, then that's really good to know, and we can kind of start to predict whether or not disease is going to be on the rise in these facilities.
Very good. That's a lot. There's a lot of uses for this data.
There are, yes. Endless possibilities with genome data.
Very good. Very good. Well, thank you so much for all the information today, Jessie.
Of course. Thank you very much for having me.
This is Sarah Mikesell with the Poultry site.